Hi, I'm Dr Marcus Keatinge, and I'm going to talk to you today at the MND LEARN event about one of my projects, which we're very excited about, which is new zebrafish models of motor neuron disease for drug discovery. So we all work in MND research, and as we all know, there are lots of challenges facing the community. The main one obviously being the lack of effective treatments for motor neuron disease. Now, this is all a complicated reason, but there are several different factors leading to this, and one of them is disease heterogeneity. So MND may present differently in different people, and also we don't really know the cause, and that may be different for all individuals. For some people, it will be completely down to their genetics. For others, it will be due to their environment. For others, a mixture. And this will all be different for different people. So not knowing a cause is also making life very difficult for us to find effective treatments. And then also, how all these interplay at the molecular level, how they finally lead to neurodegeneration, is something that's very complex with lots of different factors involved, which again is something that we're all trying to piece apart, to take this puzzle apart and put it back together. But one of the universal features of motor neuron disease is the accumulation of toxic proteins. And by building animal cellular models of disease that express these toxic proteins, we can then look into drug discovery. So what do I mean by this? So to find new treatments for motor neuron disease, we have special cell lines that develop motor neuron disease, and also we have special mice that will also develop motor neuron disease. So these will accumulate these toxic proteins that I was talking about. And what we want to do is to test potential drugs in both of these paradigms, and then essentially trying to investigate, can we use these experimental compounds to make these MND cells or these MND mouse models become healthy again? And once we identify these experimental compounds that are effective at curing the MND in these particular scenarios, we can then start to enter them into human clinical trials to find, will these drugs be effective in treating humans with motor neuron disease? But the problem is this traditional drug development process, it can be very, very long. It can be up to 10 years in length, and that's when it's successful. The way it starts off, it actually moves at quite a fast pace. So with our MND cells, we can screen tens of thousands of experimental compounds in a very, very fast time frame. So this is around about a few weeks to a few months. And when we find the best candidates from this, so it won't be every single compound, but hopefully we'd have a handful that we can then test in our mouse models of motor neuron disease. But again, we can test very few, and this will take a long time. And then we'll probably have maybe one or two experimental compounds which can then go into human clinical trials, which then may or may not be found to be effective. So one of the problems here is the fact that we can actually screen a lot of potential drugs very quickly in cells, but the bottleneck begins when we try and test them in mice. And ideally, we really want the best candidate drugs reaching the mice, which would mean that the most effective ones would then go into humans, and the more likely it is that we will find drugs that will extend human lifespan in terms of motor neuron disease. So what I'm going to talk to you about is hopefully to convince you that by using the zebrafish, we can optimize, prioritize, and then speed up this process. So just to show you how zebrafish would fit into this, as I mentioned, we have cells that we will screen drugs in really fast. You then normally go into MND mice and then into human trials. So although we can't screen tens of thousands of potential compounds in our motor neuron disease zebrafish, what we can do is screen a few hundred. So the idea would be is that you take the best few hundred drugs that come out of our cellular-based screens, test them into zebrafish, and then this would allow us to prioritize the very best compounds and triage them to go into these mouse models, which then can go into human clinical trials. This not only increases the speed, but also the efficiency of drug discovery. So why is zebrafish great as a drug discovery tool? So there is a zebrafish larvae right there. All of our testing is done in these zebrafish larvae, not in adult zebrafish, I should add. First of all, they're transparent, which makes them really great for imaging studies. They're very, very, very small as well, so you can just about see them by the naked eye. And here they are in a 96-well plate that we use traditionally for drug screening. They're also a vertebrate, which means that they have a backbone. It means they have very similar biology to humans. They have similar genes. They have similar cell types. So this means that drugs that work in the zebrafish should also work in humans. And one of the great things in terms of a screening tool is the fact that just to administer the drug, we just need to add it directly to their water and they absorb it through the skin. So it's not like with mice where you have to, invasively inject drugs, with syringes, for example. Because it's very easy to administer these drugs, it's very easy to do a lot in parallel. And this means if we can rapidly test drugs in parallel, it means that we can now do drug screening in a whole animal system. So it's all well and good saying we can do drug screening in zebrafish. If we want to do it for MND, we have to have new zebrafish models of motor neuron disease. So as I mentioned earlier, unhealthy proteins accumulate in the central nervous system of MND patients, and we can use these to model disease in animal models. One of these is TDP43, which accumulates in a toxic way in the vast majority of motor neuron disease patients. So what I did was I used genome editing to develop a motor neuron disease zebrafish, which would express unhealthy TDP43 throughout its central nervous system. We also attached a red fluorescent protein to this so we could watch it aggregate in real time. And here is a 5-day-old motor neuron disease zebrafish, and you can see the red is really concentrated in the brain and the spinal cord. So zebrafish are great for drug screening. We've made these new models of motor neuron disease zebrafish, but the question is, do they actually have MND relevant symptoms, and can we use those for drug screening? So one of the first tests that I performed on our motor neuron disease zebrafish was to look at behavior. So here we use video tracking software that will track the movement of individual zebrafish over time. And on this 96-well plate, we have individual 5-day-old zebrafish, and there's a mixture of healthy ones and a mixture of motor neuron disease ones. So if we could find a big enough deficit in their behavior, we in theory could use this as a screening tool. So when we map this out on a graph, what you can actually see, the motor neuron disease zebrafish are in the red, the healthy controls are in the black. When you can compare the two, what you see is the motor neuron disease zebrafish show a very large reduction in spontaneous movement over time. So we can actually test lots of drugs to find ones that will potentially raise this movement deficit back to the healthy levels. So this is our test number one, which we can use for screening. But also what's going on at the molecular level. So it's all well and good seeing this as an outer behavioral deficit, but what's actually going on with motor neurons? So the great thing about zebrafish, as I mentioned earlier, is the fact that we can do this live imaging because they're transparent. So here is a healthy motor neuron. And I'm going to show you a picture of a motor neuron disease motor neuron. I should add that these are the same magnification. So when we look at these nerve cells compared to the control, what you see is that our motor neuron disease nerve cell is incredibly swollen in its most distal end. So what we find is that what happens to these over time is that they get very, very swollen, they start to pull back, and then they begin to degenerate. So what we're showing is that we can get neurodegeneration in our motor neuron disease models in time-relevant time points. So this is all well and good, but how can we screen this? So I took these images under a microscope. I put the fish under there myself and positioned them. If I tried to do this with many, many fish over time, it would be very laborious, very time-consuming, and I wouldn't be very good at it. I would make mistakes. Fortunately, we have robots that can do all this for us, and we can use these to screen this phenotype over time. So this is our vertebrate automated screening technology. This special equipment allows us to automatically position the fish, image the fish, analyze the image, and it does this in a reproducible and fast way. And it allows us to look at hundreds of zebrafish throughout the course of one day. And just by taking an image of that motor neuron, what we can do is we can say, let's add drugs, and can we make those large swollen motor neurons smaller and similar to the controls. So this is just what the robot looks like when it's working in action. So here we have a 96-well plate. In each individual well is a zebrafish treated with a different compound, and the liquid handling robot will pick it up and it will feed it to the microscope. So you can see on the TV screen, there's an image of the zebrafish being rotated, and it is putting it into the identical location each time, and then the microscope will take a picture. And it can then put the fish back and then pick up the next one. So we'll do that throughout the day, looking at hundreds of fish over time. So here's just an example of some images, just to orientate yourselves. So this is a whole zebrafish, which is 4 days old. We looked at 2 different markers. These are while the fish is still alive, because the fish are transparent. And what we did was we made special glow-in-the-dark zebrafish where specific neuronal support cells are either expressing a red protein or a green protein. And the head is on the left-hand side, and then you can see the spinal cord runs all the way through the zebrafish. So just to conclude how zebrafish can fit in and speed up the motor neuron disease drug discovery process, what we can do now is screen tens of thousands of compounds in our motor neuron disease cells. We would then take the best few hundred to screen in our motor neuron disease zebrafish, so we can use our behavioral assay, we can use our imaging assay to find compounds that will make those motor neurons healthy again. We can then use our system to then prioritize the best drugs to feed into our mouse models, which will give us a better chance of finding drugs that will be successful in human clinical trials. So thank you for listening. I'd like to thank my supervisors. If anyone has any questions, they can email me. But I've had great support over the years from my supervisors, Professor Siddharthan Chandran, Professor David Lyons, and Professor Giles Hardingham, and all my sponsors. Thank you.